Welcome everyone to day 12 of Unit 3. Today we will be discussing the building and breaking of molecules. So we've discussed earlier this unit what macromolecules are, and we're going to be tying some of that information in today when we talk about how do we break down those macromolecules or how do we build up macromolecules using smaller components. Just a couple of reminders, the Elephant Toothpaste Lab is now due at the end of the day today, and next class period, tomorrow, you will have a quiz towards the second half of class. For the majority of class today, what we're going to be doing is discussing dehydration synthesis and hydrolysis. These are two processes in which we are going to be either building up or breaking down macromolecules. So we'll be discussing the difference between those two. After that, there will be some time for you to jump into Canvas and look at the study guide for Unit 3, Quiz 2. Again, that is tomorrow, so make sure you are well prepared for that. The notes that we will be covering today will be included on that. There will be just a couple of questions on dehydration synthesis and hydrolysis. The study guide that I mentioned can be found here on the main page of Canvas. So if you go to the main page, here's the agenda that I set up for you, and you scroll down to the 13th of November when we have our quiz. That study guide, again, can be found right there. You can click on that. It'll take you to a new page that has pretty much everything that is going to be included on the quiz. So notes 3.6, 3.7 on macromolecules, 3.8 and 3.11 on enzymes, and then today's notes 3.12 on dehydration synthesis and hydrolysis. So make sure you take a look at all of this stuff and review really well for the quiz. Hopefully you've already started, but in case you haven't, make sure you definitely go over some of this material before the quiz tomorrow. One other thing I want to mention before we jump into the notes is that under today's agenda for November 12th, there is an extra credit opportunity that you can complete for 5 extra points towards the summative category, and it's a Unit 3 review packet. So you want to make sure that you complete this by 5 p.m. tonight. There will be also time today if you want to work on that. It's just a comprehensive review packet of all the stuff that we've been talking about. So there's 3 pages total, and it's an all or nothing thing. So you've got to complete all of the pages here in order to get the 5 extra credit points towards your summative category. But without further ado, let's go ahead and click on the lesson agenda for today's lesson 3.12 can be found right here, Building and Breaking Molecules. The PDF notes can be found within that agenda, like always. And when you access these notes, you want to make sure that you follow along as I go through the PowerPoint and fill out all this information. To start off the notes, we're going to talk about what a monomer is. And a monomer is a building block of a larger molecule. And basically, things can be broken down into very small building blocks within biology. We have these smaller molecules that will make up larger molecules or macromolecules. So again, a monomer is a building block similar to how you have like Legos. Those individual pieces can build up multiple things. Monomers can be used to make all sorts of different structures. So we have glucose, which is a carbohydrate. That's one example of a monomer for carbohydrates. There's lots of different types of monomers for carbohydrates, not just glucose. But again, it's a different type of building block. You also have something called an amino acid. These are going to be found in proteins. There's multiple different types of amino acids. There's 20 amino acids in total. And then there's also nucleotides, which is found in DNA. So all of these are building blocks of larger molecules. And when we combine monomers together, we can actually get something called a polymer. And so a polymer is a large molecule made up of monomers. And so here I have my example of Legos again. When you take those Legos and you assemble them together into a specific shape, you actually make a lot of different things using those building blocks. And so we actually have 4 different types of monomers that will make up 4 different types of polymers. We have something called monosaccharides, and these are going to be carbohydrates, something like glucose or fructose. There's other ones as well, like sucrose, galactose, lactose, lots of different things that end in ose, and these are all sugars or simple sugars that will make up something called polysaccharides, so these complex carbohydrates. There's lots of different types of polysaccharides, like starch, for instance, is a polysaccharide, cellulose, chitin, and lots of other different types of polysaccharides that we can find that are all built from the building blocks of monosaccharides. Then we have something called amino acids, and these are going to be the monomers of proteins. Again, lots of different types of proteins that exist, lots of different types of enzymes that are basically the same thing like proteins. And so amino acids make up those proteins and enzymes, but there's lots of different variations of them and lots of different combinations of amino acids. And since we have 20 amino acids, we can make all sorts of different types of proteins from just those 20 building blocks. Nucleotides are going to be the monomers for nucleic acids like DNA and RNA. Anything that is living has genetic material, and that genetic material is made up of DNA or RNA. And that DNA and RNA is going to be unique to that individual or that species. And so using just 4 different types of nucleotides, or technically 5 if you account for RNA, out of those 5 nucleotides, we can make all sorts of different combinations for nucleic acids. And then we finally have something called fatty acids or glycerol, and those are going to be actually making something called the lipids. When they combine together, they form something called a triglyceride. And lipids are really important because when we talked about the phospholipid bilayer, that's actually made up of fatty acids and glycerol. So those monomers will help build up the cell membranes. Now, a big question that we might be asking ourselves is, where do we get these monomers to build up the organism? Well, food is made up mainly of polymers, and so when we ingest those polymers, we can actually break those down into different monomers. So we look at the label on the right here for nutrition facts of a certain food, and 3 of the 4 biomolecules can be found on this nutrition facts. We see carbohydrates, we see fats or lipids, and we see proteins. So just take a moment right here to kind of read this label and kind of predict how many of each are found in this product. All right, so from this label, we can see that the total carbohydrates is going to be 13 grams, which for a person of average size, which means 2,000 calories per day, it's going to be about 4% of all the calories that you need to ingest. So that's going to be primarily from sugars, that's 13 grams. Total amount of fats is 0 grams of fats, and then how many proteins? We've got 9 grams of proteins here. Now, if this is something that is an unprocessed food, it's definitely going to have DNA, but DNA or nucleic acid is not going to be listed under the nutrition facts. That's not something that we typically see on the nutrition facts. But just know that anytime we do eat anything that's unprocessed, it is going to contain nucleic acids. All right, so when we eat foods, enzymes help us digest them by breaking polymers down into monomers. We're taking those big macromolecules, those big polymers, and we're going to be breaking them down through mechanical and chemical means in order to break those things down into smaller pieces then that we can use to build up into something else. So for instance, we've got this image of a cat eating food. We've got mechanical digestion first. This is going to include the breaking down of the food into smaller pieces using our teeth and our jaws. Those smaller pieces of food then will go down into the stomach and they will undergo chemical digestion there. Now, the smaller you can make that food, the easier it is going to be to digest. Sometimes if you have an animal that eats way too fast, it's going to sit in the stomach a lot longer because it's going to take longer to digest. So again, the smaller the pieces that can be accomplished from mechanical digestion, the faster the chemical digestion will go. So the chemical digestion is where we have the stomach acid. The stomach acid will then help break down those molecules into much, much smaller pieces. And then somehow we need to absorb those smaller things, smaller monomers into the organism. So we're going to do that through the small and large intestines. And then, of course, there's going to be stuff that we won't be able to digest and we won't be able to actually use. So anything that we can't use or we can't digest then gets excreted out, and it's really not going to be used after that. So here's the whole process. We have ingestion, bringing in the food, digestion, then absorption, then assimilation, which is making food parts of the cell, and then finally elimination. Okay, so we're going to use all these different processes in order to break down the polymers into monomers. Polymers are then broken down into monomers by a chemical reaction called hydrolysis. So hydrolysis, if you want to break down this word in half, hydrolysis. So hydro means water, and lysis in biology means to break down. So to lyse something basically means to cut it in half. So if we combine those two terms together, water and breaking down, basically what we're doing is we're cutting something using water. So hydrolysis is a process in which one molecule of water is added to break up a polymer. So if you take a look at the example picture here, it's a little bit overwhelming at first glance, but we're actually going to take a look at it, and it's actually not too bad once you take a look at it. We've got a big molecule over here on top, and it's all joined together by all these different elements, and this is called sucrose. And so this sucrose molecule is actually an example of a carbohydrate where we have two sugars combined together. We have this hexagon and then this pentagon here. And those two things are combined together right here by this oxygen that's kind of bridging them together. And so what we're going to do is we're going to use an enzyme called sucrase to inject that water into this bond. And so what ends up happening is that we take two of these hydrogens from the water, and one hydrogen goes to the oxygen that was already here before, and then the other hydrogen, along with the leftover oxygen, goes over to that spot where the oxygen used to be. So now we have an OH group over here, and we have an OH group over here. So we've used water to break apart this molecule right here where the oxygen is. And again, we use this enzyme to do this reaction. So enzymes are used to speed up reactions. So to answer this question, where do the two hydrogen atoms and the one oxygen atoms in the water molecule go? Well, 2/3 of it, the OH group, goes to one part of the polymer, and then the hydrogen goes to the other part. So remember, enzymes are substrate specific. So each enzyme works with one substrate. For example, catalase works with hydrogen peroxide. So we did 2 labs looking at catalase. We did the elephant toothpaste lab, and then we also did the hot potato lab. And those 2 labs looked at catalase as the main enzyme to break down hydrogen peroxide. Now, catalase cannot break down everything. It could basically only break down hydrogen peroxide. And if we look at the previous slide, sucrase is the enzyme responsible for breaking down sucrose. If we were trying to break down a different sugar like lactose, lactose is something that we see in dairy products, and if you're a lactose intolerant, that means you don't have an enzyme called lactase. And lactase is responsible for breaking down lactose. So each enzyme is responsible for breaking down one specific thing. And here's a great diagram just showing you where in the human body you can find specific enzymes that will break down different macromolecules. So in the mouth, you've got salivary amylase. You've got this enzyme that helps break down simple carbohydrates. So things like bread and crackers and anything that's basically that type of material with a lot of carbohydrates. Amylase inside of your saliva can break that down into basic sugars. Then you've got hydrochloric acid and pepsin that can help break down a lot of protein in the stomach, along with lipase and amylase. That's going to help break down a lot of the fats. Sometimes that we eat foods that contain a lot of fats, that's broken down here in this lower portion of the stomach and in the upper portion of the small intestines. And then we also have lactase, maltase, all these different types of enzymes that help break down sugars further along. So all along throughout the human body, just a lot of different enzymes that work together. And here in the bottom, in the middle, you can see an example in this box of how we've introduced 4 different foods into 4 containers and introduced an enzyme into each one that will appropriately break down that food. And after a few days, that food is entirely broken down into the monomers. Now you might be asking yourself, well, if we eat food, why doesn't it take us a few days to break down that food? Why is it only a couple of hours instead of a few days? Well, you have to remember that we also do mechanical digestion in the mouth and in the jaws. So we break down those foods into smaller components that are easier to digest, and we're just basically increasing the surface area, which increases the amount of reactions that can happen, so we are able to break down those foods much faster. All right, so monomers from our food travel throughout our bloodstream to every cell in our body. So every cell in our body is connected to our bloodstream, and so when those monomers are kind of floating around, those cells can utilize that flow of monomers pretty much at any time to build whatever needs to be built inside of the cell. So there's also different enzymes in our cells that combine monomers to build polymers using our own cellular instructions. So not only do we have enzymes that will break down polymers into monomers, we also have enzymes that will build up monomers into polymers. And basically all those polymers that we ingested and broke down into monomers, that will eventually turn into stuff inside of our cells. So all our food becomes our body. So taking a look at this example here, amino acids bond to form our proteins. So our DNA actually has instructions on how to make specific proteins. And so we have the instruction manual on what proteins we need, but we just need the materials. We need each individual amino acid. So if you take a look at this protein right here, this final structure is what we want, but we need each individual building block. So you can kind of think of each circle, each colored circle as a building block. We need to have each one of those available in order to build this protein. So where do we get these building blocks? We can get them from all sorts of different sources of protein. You could have a steak, you could have beans, you could have a protein shake. All of those sources will have amino acids. Now, not everything is going to have every single type of amino acid. That's why it's good to have a good rounded diet. But every single amino acid necessary will find its way into the protein, the final structure inside of our cells. You could also apply the same logic to nucleotides to form our DNA. Same thing with lipids and with carbohydrates as well, where we can take those monomers and we build them into very specific things inside of our cells. Now, polymers are made from a chemical reaction called dehydration synthesis. So before we learned about hydrolysis, which is where we used water to cut something, but now we're going to learn about the opposite, which is dehydration synthesis. So dehydration, if you've ever thought about being dehydrated, what does that mean? It means that you don't have enough water. So dehydration means that you're losing water, and the word synthesis means to build. So in dehydration synthesis, one molecule of water is being removed in order to build the polymer. So it's basically the reverse reaction of hydrolysis. In hydrolysis, we kind of injected water into the bond in order to break it apart. But this time, we're going to take two smaller things, like two monosaccharides. We have a glucose and a glucose here. And we're going to take one OH group from this glucose and one H from another glucose. And we're going to actually take those out using another enzyme. Some enzyme is going to come into play here and take those two components out, the OH group and then this H over here. And when you pull that out, what do you end up with? Well, you end up with H2O. And H2O is just water. And it kind of floats away. And then what's left is that this oxygen that was left over, it binds to this glucose over here. So that's that oxygen over here. So where I'm pointing to now, that oxygen is the same oxygen as the one over here. So that's an example of a dehydration synthesis. And then dehydration synthesis applies to all sorts of monomers. It applies not only to carbohydrates, but it also applies to amino acids. It applies to nucleotides, everything. So dehydration synthesis and hydrolysis, they could be used to build up or break down bonds in all sorts of macromolecules. Here's an example of dehydration synthesis in lipids, where we have an OH group here and an H here. And when that gets broken apart, water will flow away. And then this oxygen becomes free to bond to something else. And that carbon has a bond that is free. So this carbon over here will bond to the oxygen over here. All right, so that pretty much wraps up our notes for day 12 of Unit 3. Again, what I would like for you to work on now is either working on that study guide, or you can take a look at that extra credit opportunity for 5 summative points.